In this presentation, I'm going to talk about how to quantify the morphology of fungal spores using ShapeGenie. To begin, let me explain how the core algorithm of ShapeGenie works. So if this is the shape you want to measure, what ShapeGenie will do is allows you to pick a grid system. In this case, it's the parallel grid system to overlay in your shape. ShapeGenie will then calculate the distance of every intersection between these black lines and your object, okay, relative to this starting point, as you can see here in step 3. And for every line that has more than one intersection, all those intersections are summed into one number, such that if you look at plot 4, there's only one value for each line position. The software then applies what's called the fast Fourier transform on the data in step 4 to get a frequency plot in step 5. And this frequency plot is a multi-dimensional representation of the shape that you began with. And if you want, you could create an index from this frequency plot by summing all bins, okay, together into one number, or just picking certain bins from this frequency plot to represent a shape. ShapeGenie gives you two choices of grid systems. At the top is the parallel grid system, which we've seen already, and the bottom is the radial grid system. They both work, and you have to figure out which one works best for your shapes. ShapeGenie offers free basic accounts so that anybody can create a free account to learn and to test ShapeGenie for themselves. We do have tutorial videos on our website, brainscreenology.com/tutorials, that will teach you how to use ShapeGenie. In this presentation, I will be analyzing images that were taken by Joseph Bartolome's, which he posted on his Facebook page. I recommend you visit his page because he takes very beautiful pictures of fungi. In this presentation, I will analyze images of spores from Cladonia ochrochloria. Here's an image of the fruiting body. Here's an image of the spores. And when I zoom into the spores, I see at least two general different shapes, right? First is this teardrop shape with a very pointy end. And then there is a rounder, a shorter, wider shape that has a blunt end. And so what is the purpose of measuring shapes very precisely? Well, in nature, when you have variations in morphology that keep recurring, these shapes indicate that there might be the presence of classes or subgroups within your population. It might be the presence of substrains, or it might indicate the presence of pathogenic infection. Also, recurring shapes might indicate certain types of environmental stressors that are affecting the organism. So for these spores, what I did, first I wanted to measure the overall shape of each spore, so I just manually traced each spore, okay, in blue ink, and then in ShapeGenie, I extracted, okay, the outlines of each of these spores. I then separated each of these spores into their individual images. But before I did any measurements, I had to first rotate, okay, each spore so that they are vertical. The longest internal line is vertical, and that the pointy end is at the top. So ShapeGenie has a tool that allows you to, okay, find the longest internal line of each shape and rotate the shape such that that line is horizontal. And then it has a tool for you to rotate the shape 90 degrees counterclockwise. Once the shape is in this form, then I request radial grid processing. But there's a little bit more detail I need to explain, because in this image, some shapes have the tapered end or the pointy end pointing to the bottom left. Or the tool that makes the shape horizontal will make these types of shapes have the pointy end to the left. So I call these the left pointing shapes. So for this category, I rotate clockwise instead of counterclockwise 90 degrees so that they also end up vertical and with the pointy end on top. I then send both of these into radial grid processing, and then I combine the results into one file. So here is a plot of just the first 10 bins of all the samples, okay, just the first 10 bins. As you can see that even in the first bin number 1, there's a lot of variation. And here in panel B, I've calculated an index in which for each sample, I summed all bins. I summed every single bin for each sample, and then I plotted the scatter here. As you can see that there is a scatter. And in panel C, I only plotted the numbers from bin 1, okay, just the number from bin 1 As you see here, there's also a scatter. So when you look at a data of a population of objects, a simple and objective way to find subgroups would be to calculate quartiles. In this case, this is quartile 1, and then this bottom blue box quartile 2, the upper blue box here is quartile 3, and up here is quartile 4. Right? And so these 4 quartiles are an objective way to define subgroups. But there's a more powerful way to do it that is called PCA, or Principal Component Analysis. And I'm going to show you data when I did PCA just on the 3 bins, the first 3 bins of every sample. Okay, so when I did PCA on the first 3 bins of every sample, I see that there are at least 3 distinct groups, right? It's pretty amazing. So in this 2-dimensional view, you see that there's at least 2 groups. You have the red group, and then the blue and green overlap. But when you look at a 3-dimensional view, you see that there's actually 3 distinct groups, right, when you do PCA. So this is the power of shape analysis. Because if you look at the original image, it's very hard to see that there are 3 groups, right? The human eye is very bad at picking out subtle shape differences. However, when you do principal component analysis, right, you see that there are 3 distinct groups. And so this is the power of shape analysis to extract insights in your research. So other shape features that might be interesting in this image of spores include subcompartments. For example, here you can trace just the tapered end or the pointy end of these spores, and I would rotate, right, these shapes to be vertically like this and use the parallel grid option, okay, in Shape Genie. An alternative is to outline the full shape of the cap and then rotate the cap to be like this, and then apply the parallel grid processing system in Shape Genie. But what about internally? As you can see, there are 2 rings in these spores. There's an outer ring, which is black, and there's a second ring inside, right, that's also black in this image. And these rings are too blurry for me to trace objectively. So what I need to do is I need to use a thresholding method. So here I use a software called Fiji or ImageJ. And what I did was I cropped out the inner part of the spore so that the only thing left is these 2 rings. And then next I turned this image into grayscale, right, such that our rings become dark black. And then I inverted the intensity such that our rings become bright white. And then in step 4, I filled in the black hole with gray. And in step 5, I thresholded for brightness, which allowed me to create a mask of all the bright pixels. And in step 6, I extracted a mask, right, which is a mask of what? The rim that we're interested in, these double rings. And then in step 7, I would then rotate this object vertically like this with the opening at the top. And then I would choose the radial grid processing system. You see that? So it is possible to isolate the rim from these spores and to do it objectively by thresholding as opposed to manual tracing. What about the inside of the spore? We can also isolate the inner part. As you can see here, there is no clear shape on the inside of the spore, but you can see texture. That is, it looks bumpy, right? There are bright spots and dark spots. And so what I would do is I would crop out the inner part of the spore to get rid of the rim, and then I would turn that to grayscale, and then I would threshold for brightness, and that would allow me to create a mask, okay, of the texture within the spore. I would then rotate this to be vertical, and then I would apply the parallel grid LCPC system, okay, in Shape Genie. So you see, even texture can have a shape. I like to conclude by saying that in biology, the mantra is structure is function. That is, when you see recurring patterns of distinct shapes, these patterns will correlate with distinct functions. Why is this relevant? Well, recurring patterns will often correlate with features that give insight into your biology. That is, differences in soil temperature and moisture, such as due to droughts or floods, right? Differences in moisture in the air due to seasons, right? Prolonged hot seasons, the presence of pathogens that are infecting your organism, the presence of pollution, right? The presence of genetic variants or substrains or subspecies. Shape analysis can help you extract patterns that then correlate with these features to help you better understand the biology that's going on.